Hey, A&Pers, this is Dr. Jones checking in. This is a video lecture for A&P 2, lecture test 2, video 8. It says 8. And we are starting on the top of page 19 with the pancreas, which looks like this, if your page numbers are different. Actually, no, it's not 19. I said 19, and then look what's in the corner. That's a 20. Did we do 19 on the previous video? Yep, we talked about the adrenal medulla, epinephrine, the pinealocytes, melatonin. Okay, we're on page 20. Let me fix that on my... I'm writing the page numbers down so that I can put them in the description on the YouTube video and on your Canvas so that you'll know which video to go to when you're trying to fill in a certain page on your note set. So we are at the pancreas. The pancreas is both exocrine gland and an endocrine gland. It is a mixed gland because it makes both secretions and hormones. Now the exocrine portion of the pancreas we will come back to and talk about in digestion because it makes digestive juices, which as a group are going to be called pancreatic juice, which is weird. But it does actually the majority of your digestion. So, and that's 99% of the pancreas is the exocrine pancreas. And then, wait, where's the pen? Does that work? And the endocrine pancreas is 1% of your pancreas, and that is the part that makes hormones. And it is pancreatic islet, this slightly lighter red-ish pink splotch in the pancreas is the pancreatic islet. They used to be called islets of Langerhans, and it is a silent S like the word island. Oh yeah, the word island has a silent S. You've been saying it this whole time, but now you know. You're welcome. So there are 4 types of cells in the pancreatic islet. For the current lab list, it just says, oh look, they're islet cells. But for lecture, we need to know the different types of cells and what they do. So the alpha cells make glucagon, G-L-U-C-A-G-O-N, glucagon. And we'll talk about that at the top of the next page and what it does. The beta cells make insulin. You've heard of insulin. The delta cells make the growth hormone inhibiting hormone. Also known as somatostatin. And probably should tell you what each of these do. Okay, so the alpha cells that make glucagon functions to increase blood glucose levels by breaking down glycogen and releasing glucose from the liver. The beta cells that make insulin functions to decrease blood glucose levels by moving the glucose into the cells and making glycogen. Glycogen, again, was a sugar storage molecule. The delta cells with their GHIH, the somatostatin, slows the release of glucagon and insulin, so it's inhibitory, IH, to both the alpha cells and the beta cells, both the glucagon and the insulin. And the PP cells, that's right, I said PP. And it stands for pancreatic polypeptide, although there's clearly 1, 2, 3, 4 P. So PP. No. But no. And they inhibit gallbladder contractions. And gallbladder contractions are very important. We'll talk about the gallbladder when we get to digestion. But long story short, basic concepts. The liver makes bile salts, And the bile is stored and concentrated in the gallbladder. And then when you eat fats, and those fats reach the beginning of the small intestine, it causes contractions of the gallbladder that squeeze the bile salts out into the small intestine to emulsify fats, which is to break the fats down into smaller bits. So the pancreatic polypeptide cells inhibit gallbladder contractions. Okay. And at the bottom of the page, we have glycogen, C-O-G-E-N. We've got a number of words here that all sound and look a lot the same. If you're a whole word reader, then glycogen and glucagon, G-L-U-C-A-G-O-N, glycogen and glucagon both start with a G, end with an N, have an L, have a G, vowels. It's like horse and house. If you're not reading every letter, it's the same word. The shape is the same. So glycogen, we have to be very careful here to know that glycogen is the sugar storage molecule found in liver and skeletal muscle cells, whereas glucagon is a hormone that raises blood sugar by breaking down glycogen and releasing that sugar into the bloodstream. Okay. Let's talk about insulin in more detail. Okay. And insulin is a peptide hormone released by the beta cells. Okay, just making sure my father-in-law wasn't going to trip over the dog. We're good. And glucose is high. Okay, insulin is a peptide hormone released by the beta cells when glucose concentrations are high. Normal is 70 to 110 milligrams per deciliter at this time. So there aren't a whole lot of numbers that I ask you to memorize in this class, either ANP1 or ANP2. And anytime we do come across a number I want you to memorize, I specifically tell you, and I am telling you, memorize this number. So normal blood sugar range, 70 to 110 milligrams per deciliter. Insulin enhances glucose absorption and use. The previous version of the book said utilization. It's the same as use. And target cells are insulin dependent. We could list the insulin dependent cells, but we would be here all day. So let's take a shortcut and list the insulin independent cells. So this is a list of cells that don't use insulin. Brain cells, kidney cells, digestive tract lining, and red blood cells. Most everything else is insulin dependent. Okay. The effects of insulin. They accelerate glucose uptake into the cell. So insulin is released, here's our cell, when the blood sugar levels are high. Lots of glucose in the blood sugar, in the bloodstream. So what insulin does is it causes the blood sugar to move into the cells. Excuse me. Excuse me. Okay, and it makes your body use more glucose and enhance ATP production. So the sugar goes into the cells and your mitochondria use it to make ATP. That one didn't look like a mitochondrion. It looks like an angry face. It looks like an angry cyclops with bad hair. Wow, that's spectacularly terrible drawing. I apologize. Okay. It stimulates glycogen formation. Glycogen, again, was our sugar storage molecule found in the liver and skeletal muscle cells. Stimulates amino acid absorption and protein synthesis. So again, amino acids absorption means putting them into the cells. in protein synthesis, so we're going to use these amino acids to make proteins. And stimulates triglyceride formation. So the function of insulin is to get the glucose into the cell. And when do you have high blood sugar levels? Usually that'll be postprandial. Postprandial. Y'all know that term? P-R-A-N-D-I-A-L? I-E-L? Okay, one or the other. Postprandial means after a meal. So after you have eaten and then had some ice cream, or 44-ounce big gulp with 6 different sugary syrups in it. Yeah, I recommend against that. You'll have high blood sugar. So after a meal, you get those sugars out of the bloodstream and into the cell, and you store them to make glycogen. Here's your sugar storage molecule. It's what it looks like. It's a bunch of glucoses just pop beaded together, but like a branched pop bead structure if that were possible. And also, again, after a meal, get those amino acids into the cell, use them to make proteins, get those fats out of the bloodstream and store them as triglycerides, get the excess sugar and store it as glycogen. So this is a postprandial hormone that helps you digest, because it helps you use the digested materials that are in the bloodstream. It gets them into the cells, stores the excess, it makes ATP that you need, and stores the excess. Okay. Need to know. Next page. Okay, we'll come back to that. All right, so the insulin, the beta cells make insulin. It does everything we just said, and that lowers the blood glucose levels, bringing you back to normal blood glucose. So when the glucose is gone, release glucagon. Get it? When the glucose is gone, release glucagon. So when blood glucose concentrations are low, and also you see those brackets around blood glucose? That's the chemistry abbreviation for the word concentration, which I no longer spell out. When blood glucose concentrations are low, the alpha cells release glucagon to mobilize energy reserves. So it does kind of the opposite of, it does the opposite of insulin. Not kind of the opposite, it does the opposite of insulin. Primary effects break down that stored sugar in the skeletal muscles and the liver cells. Break down the stored triglycerides. I didn't do the timer again. And break down, start making glucose in the liver, which is gluco for glucose, neo for new, and genesis for make, or start, or beginning. So start to make new sugar, gluconeogenesis, when you make new glucose. As you recall, your brain only eats glucose, and when you're out of glucose, when the sugar is gone, release glucagon, you can actually make new glucose in the liver. Fun, right? And the results are a reduction in glucose use, and release of more glucose into the bloodstream, causing blood glucose concentrations to rise towards normal. Towards. Not an English major. Parasympathetic stimulation enhances insulin release, while sympathetic stimulation inhibits it. You would think, because insulin makes you make more ATP, that would be sympathetic, but this one makes more glucose. And remember, insulin is at the tail end of your digestion, so it's a rest and digest parasympathetic thing. Glucagon is sympathetic. Okay, what's this next slide? Okay, so here's this. When blood glucose levels are gone, you release glucagon, which causes breakdown of glycogen to glucose, breakdown of fatty acids, and make new glucose, and that increases blood glucose levels. Diabetes mellitus is high glucose concentrations high enough to overwhelm the kidneys. Hyperglycemia is high blood glucose levels. Glycosuria is glucose in the urine, urea, glucose. Polyuria is too much urine. All of the diabetes will have polyuria, both diabetes insipidus and diabetes mellitus. Too much urine is diabetes. Type 1 diabetes is where the pancreas does not make enough insulin. This is also called insulin-dependent diabetes, where you need to take shots or like have the insulin pump with a constant influx of insulin because you don't make any or don't make enough. Type 2 diabetes is insulin resistance, when you make insulin but the cells don't respond to it. So this can at the beginning just be downregulation, or maybe the receptors don't work. I mean, there's lots of things that can happen. And diabetic nephropathy, a nephron is the basic unit of the kidney. So, and pathy is a pathogenic, something has gone wrong. So diabetic nephropathy is when diabetes mellitus does damage to the kidneys. Diabetic retinopathy, you know where the retina is? It's in the eyeball. So this is diabetic damage to the eyeball. And diabetes can eventually cause blindness slowly over time. And diabetic neuropathy, peripheral nerve disorders, generally the feet first and then the hands. So there's that. Okay, and we only have 3 pages of Noteset left. Think we can do that one more video? Okay, well, I did not put the timer on, and did we do 2 pages? Yeah, we did 2 pages. We've been doing 15, 16, mostly 2 pages, occasionally 3 pages, and we've got 1 page left, and these are all secondary endocrine functions, so we'll put them together in one video. So this has been MP2 Lecture Test 2 Video 8, Noteset pages 20 and 21. Thank you as always for tuning in. The education must go on. Dr. J out. Control, Command, Escape. Control, Command, Escape. Have a good one.